Thank you, Ed. So I'll be talking on my dissertation process of flathead catfish, Pylodictis olivaris. So flathead catfish are a long-lived species that are both recreationally and commercially important. They're found throughout the Mississippi River drainage basin and into most of Texas, most of Alabama, and most of Mississippi. They are a habitat generalist and occupy a variety of habitats, be it in either moving or still water ecosystems. They are a lion weight piscivore, and they can feed on a variety of fish, be it clupeids or centrarchids. It's also a highly fecund species, with females producing many tens of thousands of eggs, and they exhibit age-specific fecundity. So they mature around 3 to 5 years of age, and they reproduce with individuals building and guarding nests. Now, currently in the state of Missouri, there is a lack of trophy individuals. Let's just say individuals greater than 1,020 millimeters, so around 45 inches. And this could be due to three possible reasons, be it high competition rate for food and shelter, high angling pressure, or illegal harvest through methods such as hand fishing, where anglers are, say, trying to induce a defensive response from catfish on these nests by sticking their hand or arm in there. And it's gone so far as anglers placing structures in the water column here to actually provide spawning habitat for flathead catfish, and Missouri Department of Conservation has subsequently removed those in this bottom picture. MDC has introduced a statewide bag limit of 5 fish per day, but not all reservoirs have the same bag limit. Some have more, with you allowing to keep, say, 10 fish per day. There's no length limit in place statewide, though again, some of the smaller reservoirs, they differ. They actually have a 24-inch minimum length limit. And again, noodling or hand fishing is a legal practice. So if we want to see then what could possibly be happening for why we don't see as many trophy individuals, we could use traditional fisheries metrics such as weight, length, etc., length distributions, or what I did, which was I used genetic markers. Specifically, I developed microsatellite markers in my first chapter. So what are microsatellites? Well, very simply, they're just single sequence repeats that can be tens of 100 base pairs long. They can be 1 to 6 base pair repeats, and these are regions that are highly susceptible to mutations across the genome. The reason being is because we have a large number of repeats, when you're going through the transcription phase, you could have some slip strands, some issues that are regarding the transcription that results in the expansion or truncation of that repeat area. So this means then that these markers and these areas are typically highly polymorphic and also very abundant and widely dispersed markers. Now, if I'm going to use microsatellites, we got to make two assumptions. First, that those loci are always going to be unlinked. And second, you have to choose between models. And there are two models roughly. We have the infinite alleles model, where each individual mutation results in a new allele, or what I used was the stepwise mutation model. So if we take a look, each mutation can then create a new allele, but say here within this area, we have mutations that results in an allele that's already been present in the population potentially. This is the model that I utilized for my markers. The other reason that I was looking at microsatellites is it's a codominant Mendelian marker. So that means, say I want to go through and I run and do my PCR and I'm looking at my results. Since it's codominant, I will see possibly both alleles if that individual is heterozygous at that particular loci. One of the issues, though, that's been historically prevalent with using genetic techniques is it's difficult to locate and generate these markers just due to cost of DNA sequencing, as well as the amount of labor that was required. But with the advent of high throughput sequencing technologies, such as this Illumina HiSeq platform, that cost and time has decreased. Now, within ictalurids, microsatellite sets have already been developed for species such as channel catfish and several species of mad tom. While I was conducting the actual initial sampling period and as well as identification step, White et al. Published a set as well for olivaris, though this was in a population in Pennsylvania, which was invasive, and I wanted to have one that was native within the olivaris range. So I needed 3 criteria for my microsatellite loci. First, they just need to be reliably amplifiable. Anytime that I'm going through and doing my PCR, I don't want to be wasting time. They had to have multiple alleles present, meaning it's not fixed for one allele because it's not very informative. And it had to conform to Hardy-Weinberg expectations. So my objectives for this study were pretty simple. Just find 15 microsatellite loci in this chapter. How I did this was I took one individual that was collected during an initial pilot sampling event during the spring and summer of 2020, and I extracted tissue from that individual using the D&EZ blood and tissue kit. The recovered DNA from that individual was sequenced on an Illumina MiSeq platform, just a tier below the HiSeq, in that it's not producing as much data as the HiSeq. And in terms of number of reads, the recovered sequences were then input into the QDD pipeline. Within that particular pipeline, I gave preference to repeats that consisted of di, tri, and tetranucleotide repeats, so 2, 3, and 4 repeats. And the design primers then underwent testing for PCR conditions. So this was two things. First, figuring out multiplex combos. These markers are like kids. Some work together, some don't. Some inhibit others, while others work just fine. The other was focusing on annealing temperatures. They are particular with when the primer's designed, there's a particular annealing temp that it is optimized for. So figuring out those conditions was then the next step. Once I collected my genetic data, I then conducted some simple analyses. First, using GenLX, I looked at the number of alleles that were present for each loci in each population and the expected versus observed heterozygosity estimate. So here, if I had more observed than expected heterozygotes, I had an excess of heterozygosity, and vice versa for an excess of homozygosity. I also used GenePop to conduct some exact tests for deviations from Hardy-Weinberg, and I also used Pop Gen Report to conduct one more genetic diversity estimate of allelic richness. So here, I then took that estimate and used a Spearman rank correlation just to see if there were any differences between reservoir size and the allelic richness value. So my hypothesis rendered around that as we increase population size, it's a larger population, larger from a larger lake, I should have more alleles and a higher value present. Move to results. So out of 150 tests, 34 were significant across all reservoirs. 33 of them had an excess of homozygosity at those particular loci. Though I actually had one that had an excess of homozygosity at pole 31 in Higginsville City Reservoir. For loci 15, 27, and 33, they had the most tests for excessive homozygosity at those loci across all reservoirs. If we will break them up into reservoir size, for large reservoirs, Harry S. Truman had the most tests significant at 7 between the two lakes. For medium reservoirs from 5,000 to 40,000 acres, Stockton had the most tests significant at 4. And for the small reservoirs, Higginsville City Lake had the most loci out of significant tests at 5. What I found for reservoirs greater than 5,000 acres, 14 out of the 50 tests were significant, and this is more than likely due to some population structure that was present. If we look at the correlation, I found that as the reservoir size increased, I saw an increase in my overall allelic richness value. So larger population meant there were possibly more alleles present in those populations. So some conclusion I can draw is, again, allelic richness increased with reservoir size. I found, for the most part, 12 usable microsatellite markers that I could use for my downstream analyses. And again, most of the tests were in the medium and large size reservoirs, more than likely due to the Walloon effect. So as I saw some subpopulation, there were some alleles present in those subpopulations that weren't in others that drove that particular result. So the markers that I found, I then used for all my downstream analyses to then estimate my population structure and effective population size for olivaris in these reservoirs. Now, why population structure and effective population size? We've seen that as humans have gone about, we are fragmenting populations across the landscape. If we think about it in terrestrial ecosystems, urbanization creates smaller habitat patches, and this is no different in our waters. We can take example Harry S. Truman, which serves as a nice, easy, definable barrier to movement potentially for fishes within rainwater drainage basins. But we could easily see it at a smaller scale with culverts as well for more smaller bodied fishes such as minnows and darters. Now, one reason that I'm focusing on genetic diversity is because it's going to impact the overall fitness. So how easily genes and alleles are passed on to the next generation, as well as the evolutionary potential of populations and species. And Genetic diversity is important because it's going to be lost due to processes such as genetic drift and inbreeding. And therefore, measuring them is going to be very important for overall conservation efforts. Now, effective population size is the first thing that I measured. How does it differ from total population size? Total population size is simply the number of individuals that are present. Just go out, count the number of individuals, that's your N. NE is going to be the size of the ideal population with the same amount of genetic drift as the natural population. So it serves as a measure of genetic drift in the population. So what we typically see is that effective population is then typically much more smaller than the overall population size. Why this matters for the conservation efforts is the idea of this IUCN guideline, the following the 50 to 500 rule. So if we want to maintain genetic diversity in our populations and we have a short-term conservation goal, we need at least 50 individuals to maintain that genetic diversity. If we are looking for a longer-term goal, we need an NE of at least 500 to maintain that diversity. Now, how I go about measuring NE, we have three possible methods we could use. First is the temporal method, where we are just sampling a population from 2 points in time, and we're just measuring then the changes in allele frequency. This is a little bit impractical for my species because it can live up to 30 years. So that means if I want to come back and do this, I have to wait 30 years, which is not really practical. The other option I have now is linkage method. So we've changed it. Now I'm just measuring the non-random association of alleles that's going to be present within the population. Here I got to assume one thing, and that's the loci are independent and drift is the only thing that's responsible for the signal that I am getting. Benefits here is that it only requires a single sample from the entire population. So this is the method that I used for this chapter. The last one is the parent assignment. So this is analogous to marker capture sampling. I'm measuring the overall reproductive variance of a particular population. Here, though, we got to assume two things. First, that the population size is going to remain constant in time. And second, that any sort of age structure is going to be the result of random mating. So very little sexual selection, as well as random births and deaths. This was utilized in my third chapter, which we'll see later. The last thing we'll define is population structure. So here, population structure is very simple. It's just allelic differences between subpopulations. And this is going to occur as a result of limited gene flow. And if we take an example of my home area in the Gulf of Mexico for black drum, red drum, and spotted sea trout, we could see population structure occurring in possibly two ways. One could be isolation by distance, whereby as we increase distance, we see increased genetic differences between these subpopulations. One example that we can draw is, say we have a population that is down in Brownsville. It's going to be quite distinct maybe from one over in near New Orleans, because that is several thousand kilometers away that fish would have to travel or in order to breed and to maintain that gene flow. The other possible reason could be we have a barrier to movement. So we could say maybe the Mississippi River, which is going to have a very large change in salinity, could provide a barrier to movement. We've seen that from other species within the Gulf as well, where we have some species in the west that we don't see in the east and vice versa. So the consequences then of this is that, well, these subpopulations are more susceptible to genetic drift and inbreeding, which then means the population could have a greater extinction risk. So my objectives then for this part of the study were to assess the populations for any genetic structure and estimate the overall effective population size within these reservoirs for olivaris. How I did this was, first we had to figure out how many samples and numbers of genotyped individuals I'd need to actually ascertain confident estimates of NE. So I used the program NeoGen to do this. It requires two sets of input parameters. First is population demographic parameters. So things such as minimum mating age, maximum age, natural mortality rates. These are going to be parameters that have been previously estimated from research in olivaris, as well as any data that have been collected by the Missouri Department of Conservation. I then also utilized population genetic parameters, which were collected from my initial sampling and microsatellite discovery time. So that included things such as mean allele locus and alleles per locus standard deviation. I then ran sampling scenarios for each reservoir class in this study. So now we actually know how many samples we roughly need. It was time to actually go do the sampling. So sampling occurred from 2020 to 2023. This occurred in the spawning season, so late spring to early summer. I had 10 reservoirs. If we look in this lilac color, these are small reservoirs. This green is going to be medium, and the papaya is large. Within each reservoir, 20 to 50 sites were selected for sampling, and I used 15 hertz on a 30% duty cycle electrofishing, along with the other MDC biologists that used it. How this manifested was in large reservoirs. There was a two-boat strategy, a main boat that remained stationary and sent the electronic pulses into the water column for 3 to 5 minutes, and then a chase boat that moved around and collected individuals that surfaced further away from the main boat. In small reservoirs, this was a one-boat strategy, so there was one boat sending out the electronic pulses that then was free to move about and collect individuals as they surfaced. Once each individual was collected, a weight and total length measurement were taken, and a fin clip was taken from the upper caudal lobe. That fin clip then was my genetic data that I extracted the DNA from and then ran from my microsatellite loci. Any of the recaptured individuals were then used to estimate the overall population size within these reservoirs. So I have my genetic data. Now it's time to actually start doing some analyses. So for my population structure, using the GenAlX program, I calculated AMOVA to detect any hierarchical partitioning. I estimated an FST value for each reservoir and then conducted a principal's coordinates analysis. I also used structure in combination with this to just determine the appropriate number of genetic clusters that might be present. How I did this is I ran it from K 1 to 10, and within each K, I ran it 10 times. So 10, 1, 10, 2. 10 times I ran 1, 10 times I ran 2, with an initial period, burn-in period of 50,000 steps, and then a 200,000 NCMC step period. The results I recovered there, I then analyzed using structure harvester, and then if I need to visualize them, I use clump and destruct. For my effective population size estimate, I calculated that for each population and subpopulation I detected an NE estimator. If I did detect subpopulations in these reservoirs, I attempted to get an estimate for the overall reservoir using the Hololey et al. Methods. So let's move to results. So in small reservoirs where it is estimated I'd have an effective population size less than 100, I required at least 60 genotyped individuals to get confident estimates of effective population size. So this gray bar here represents the true value. And as we increased the number of genotyped individuals, the closer that value was to the actual true value of NE. And I also saw that my confidence intervals became much more narrower, so I was much more confident in the actual estimate that I recovered. In medium reservoirs and large reservoirs where the NE estimate would be greater than 100, I required at least 100 genotyped individuals. And we can see this again. The red represents estimates that were not even appropriate estimates of NE for the program. But until we reach 90, now I have an estimate, but I don't have a very confident interval. And it was after 90 individuals were genotyped that I then recovered that estimate of narrow confidence intervals with high confidence and an actual NE value that we were pretty certain was the true value. All right, so we'll now actually get to the meat of the actual results. So spoiler, all the small reservoirs had no population structure detected. K equals 1 had the highest likelihood for these reservoirs. So all my NE estimates were for the entire reservoir. So in Bilby Ranch, that estimate was 28.5. For Cameron, that estimate was 35.2. For Cheru, that estimate was 6,587. And then for Higginsville City Lake, that was 76.6. Finally, for Long Branch, it was 275.7. Now we'll move to the medium reservoirs. So here we're going to first focus on Pomme de Terre. Focus on the right-hand side, the PCOA analysis. We see a nice shotgun blast pattern with very few individuals that are kind of moving further away from the rest of that cluster, showing very little population structure to no population structure. My FST value here was 0.003, so roughly about 0% of the variation was because of between the populations. And K equals 1 had the highest probability for this reservoir from structure. As a result, I calculated my effective population size for the whole reservoir, and that estimate was 868.1. Next, we'll move to Smithville Reservoir. Again, we look at the PCOA first. We see it's a nice shotgun blast pattern up top, so very little differentiation on the plot. My FST value here was 0.01, so 1% of the variation was between the populations. In order to say that there is some subpopulations present, I needed around at least like 5%. And again, structure had K equals 1 as the highest likelihood for this reservoir. So again, the NE estimate was for the entire reservoir, and that was 128.3. Finally, let's look at Stockton Reservoir for the medium reservoirs. Here, if we actually look at the PCOA, we do see some shifting away between these two reservoir arms. And I had an FST value of 0.03, so about 3% of the variation was due to between the populations. However, structure, though, had K equals 1 as one of the highest likelihood for this reservoir. So even though I have a decent amount of variance that's present, I still calculated the overall NE estimate for the reservoir. And that estimate was 516.9. All right, so we'll move now to the large reservoirs. These are the last size class. First, let's look at Table Rock. If we look at the PCOA, we actually see that there is some distribution away of individuals in Long Creek from the rest of the reservoir. Those are individuals that are going to be present in the southeastern corner. I recovered the highest FST value within my study here at this particular reservoir. It was 0.085, so about 8.5% of the variation was between the populations. And on my structure plot, K equals 3 had the highest likelihood for this particular reservoir. But if we actually look at the structure plot, I determined that K equals 2 best fit the data. Each bar represents one individual, and again, it was all the individuals in this very southeastern corner, these yellow circles, that are differentiating away from the rest of the reservoir. So with that in mind, I then tried to see if there was any population structure present in the rest of the reservoir that was being swamped out by those from Long Creek. So I reran it. Under the PCOA analysis, we see again a nice shotgun blast pattern. And again, an FST value that was very, very low of 0.002, so about 0% of the variation. So I then just estimated the effective population size for two subpopulations. For Long Creek, that estimate was infinite. For the rest of the reservoir, the estimate was 1,499.4. And for the overall reservoir, I attempted to try to use the methods from Holly. However, for this reservoir, I got an estimate of infinite. Lastly, let's look at Harry S. Truman Reservoir. We look again and one more time at the PCOA for the whole reservoir. We see a nice shotgun blast pattern, very little shifting away of individuals or any subpopulations. My FST value here was 0.02, so only 2% of the variation was among the populations. Structure here actually recovered, though, K equals 2 with the highest likelihood within this reservoir. And what I found was that I had individuals in the southern area that were differentiating away from those in the northern area. So with that in mind, of course I reran it. So I reran it for individuals in the northern area, the South Grand Tebow Creek region. PCOA, very little differentiation there on the PCOA, and I had an FST value of 0.003. So only one cluster here. If we go to the southern half, this Osage Palm de Terre area, again, looking at the PCOA, very little clustering away from each other, just a nice grouped pattern. And the FST value was 0.009, so about 1% of the variation. So determined then just one cluster. So my NE estimates then were first for the Osage Palm de Terre, this southern cluster down here, was 121. For the South Grand Tebow Creek region on the northern half, the estimate was 224.5. This time, though, I actually got the Holly methods to provide me an estimate, and that estimate was 352.55. So let's put a quick bow on this. For small reservoirs, no population structure was present, and for the most part, I recovered credible NE values. The lone exception is going to be the Che-Rue Reservoir. More than likely, that's just a result of not enough individuals being genotyped for that reservoir to get a good estimate. And this is more than likely just due to the actual logistics of getting the samples, having a truncated season to do so, as well as also having to try to sample 10 other reservoirs. And if we take a look, for the most part, these smaller reservoirs are having that NE value that's below the 50, so below that guideline. So it's possible that they might be a conservation concern, but it also begs the question of whether or not they could support a larger population as well. For the medium reservoirs, slight population structure was detected, but I did at least get credible NE values for those particular reservoirs. And finally, if we focus on the large reservoirs, there was obvious population structure that was present. I did not recover an estimate for the Table Rock Reservoir. More than likely, this might be due to the fact that that reservoir is so large and supports a population so large, no number of genotyped individuals could give me an estimate there. All right, so now we got to cover quickly, though, why I might be seeing possible reasons for that obvious structure. So let's first focus on Table Rock. More than likely, this might be a case of isolation by distance. If we look at where they were collected and then the closest individuals, that's about roughly 11 to 12 kilometers away as the crow flies. If we actually include then where they would have to travel via the water column, we're now starting to get towards 15 to 20 kilometers. So that's about a little over 9 miles to start with. So if they are having optimal habitat present, they might not be willing to make these long distance dispersion events to then to the rest of this reservoir. For Harrius Truman, there's a possibility that this is actually a little bit different. This might be more a sign of site fidelity by different groups in different life stages. So what I found is that these blue squares were mainly smaller, early mature, or juveniles, and the yellow circles were older individuals. So these older individuals had made the long distance migration or movement patterns down to the southern area and were focused around timbered areas that are going to support more of that piscivorous diet. So those larger clupeids such as gizzard shad, but also centrarchids. Whereas the younger individuals were around more riprap. So they were more focused on preying on crayfish and other smaller piscivorous fish, such as threadfin shads. So we've seen that there is population structure. There is some degree that needs to be conserved. How do changes then in management practices affect them? And that formed the basis of my third chapter. And this is extremely important because the goal of fisheries agencies is to implement management strategies to create not only sustainable, but preferably quality-sized individuals for anglers to capture. And what has been historically been utilized is traditional fisheries sampling techniques or sampling data, where we're looking at length, weight, length frequency histograms to understand when a population might be potentially overharvested and modeling at that. But the advent of genetic techniques has allowed us to estimate parameters that could allow us to have a little bit more refined view on when a population could be close to overharvest. And this is especially important for these populations because angling is adding an extra layer of mortality on top of the overall natural mortality they're already experiencing. As well as angling tends to remove larger individuals. We all want to go out, we all want to capture the biggest fish we possibly can. And this can lead to detrimental impacts if we focus on females, because females tend to produce better quality size and more eggs if they are larger under this big old fat female fish hypothesis. And we can even see that from this example here with red snapper, where if we're looking at around 1962, we had a prevalence of individuals that were quite large, 33 inches in length, and quite old for this population, producing lots of eggs. But as these individuals were fished out, what we now see more currently today is fish that are maturing earlier, they're smaller, they're producing fewer eggs. And this could have detrimental impacts on the overall fecundity of the population. And through instances such as that, agencies have implemented strategies to minimize the impact that angling length has, be it through actual length or bag limits or just pure education. Now, I mentioned length and bag limits. Let's go over them quick. So bag limits just help to say you're only allowed to take certain numbers of fish. So we're limiting the overall mortality of fish. So if we have this group of 8 catfish where historically you could remove as many as you want, say we put a limitation where you can only take 2, we still have 6 individuals that could be potential breeders that could pass their genes on to the next generation. Whereas size limits, we are trying to limit the impact of angling mortality on a specific size group. So first we have minimum size limits where we're trying to protect the smaller individuals in a population. We're allowing for these younger individuals to mature and then reproduce before being subjected to that angling mortality and possible harvest. We could use maximum size limits where we are wanting to protect these larger, more highly fecund individuals so we don't lower the overall fecundity of a population. Or we could have slot length limits where we are just trying to protect one certain size range while we compromise and allow anglers to target the smaller and larger individuals within a given population. So my objectives then were threefold for this particular part of the study. First, assess the reproductive variance of olivaris in a reservoir. Second, model the length and length limits and fishing mortality effects on total population size, effective population size to total population size ratio, NE to N ratio, and the generation length. The third thing was just model the effects of these length limits and exploitation on the size distribution of a population. So how I did this was we selected one reservoir and just sampled it extensively. That reservoir was Higginsville City Lake. Overall goal was to collect as many individuals as possible. Once that individual was collected, they were attempted to be identified as either male, female, or immature. Alongside the weight and length measurement taken, along with the fin clip that was taken for the genetic data, a pectoral spine was removed to estimate the age of that particular individual. Those that were captured and then I received the fin clips for, I genotyped for all my microsatellite loci to then first conduct a parentage analysis and service. So here this was occurring in 2 steps. An initial simulation period based on the manual's recommendations. I ran it where I simulated 100,000 offspring. I only used adults greater than 3 years old. Those would be the individuals that would be maturing. The proportion sampled I had to specify and I set for 0.66. The reason for this was I used the number of genotyped individuals that I had divided by the Chapman mark recapture estimate that Missouri Department of Conservation provided me. I also used the likelihood of the distribution for my confidence estimates with 2 confidence level estimates, a relaxed and strict, to then assess out the actual likelihood that I was recovering for that simulation. And then the second step was the actual parentage analysis, time to figure out who the parents are. So here I used the simulations with the highest likelihood score first to actually run the analysis. And then I used all individuals that were captured as potential offspring. And again, only the individuals greater than 3 years old were considered possible parents. For each offspring, the output I chose was the 2 most likely parent options. This was due to the fact that if I, say, got 2 individuals that are more than likely siblings, I then had a potential parent underneath that I could then evaluate if that was the actual parent or not. I ran this analysis separate for males and females, so I did it in 2 runs, 1 for males, 1 for females. I also conducted a SIP-SHIP analysis utilizing 2 programs. The first was ML-Relate, where I was looking at the overall likelihood 2 individuals were related. So here the microsatellite data was just the input file, and I used the specific hypothesis testing option to see if individuals had possible 4 relationships. Either they were parent offspring, full sibling, half sibling, or unrelated. I also used Colony 2. So here, instead of just pure likelihood, I used a full and pair likelihood analysis. And I was able to have a few more input parameters before conducting the analysis. So under my mating system, I allowed males and females to be polygamous, and I allowed inbreeding. I also selected for the alleles to be calculated, but not updated. The reason being, if I allowed the alleles to be updated, I am assuming I understand something around olivaris family structures, and at this moment in time, I did not. I allowed all offspring to be individuals to be possible offspring, and again, only those individuals greater than 3 years old were possible parents. At the conclusion of that SIP-SHIP analysis, I took the output relationships from both programs, compared them to see where they overlapped for the number of full and half sibling relationships. Why I did this was I used equation 4 from Waples and Faraday to provide an estimate of NE. So here I'm using this NE estimate to assess the overall success of the relationship and compare it back with the estimate that are covered in chapter 2. So if the values were closer to each other, I determined that the analysis was an overall success. Finally, to actually assess what the effects of management decisions would be, I used the program Age-NE. This is a program from Waples where he has generated several equations and has input them in towards a program where I could then model the effects, different length limits or exploitation rates. And it allowed me to focus on parameters such as total population size, effective population size, that NE to N ratio, where I could see like if any reductions were occurring, that there's going to be a decrease in the overdispersion of reproductive variants, as well as generation length, which allows me to look at the average time between two generations in a population. So say our average generation length is increasing, there are more older individuals that are reproducing in that population. And as well as size distribution was another output. Now, for this program, it only requires a handful of parameters. The first is age. The next is age-specific survival rate. That I took from Winters and McMullen's previous study on their estimate of natural mortality. I also broke up my ages into specific size classes based on Daugherty and Sutton to look at the size distribution. Here, if we take a look at the table on the right, how I broke it down was I had individuals 0 to 350 millimeters, which corresponded roughly to 0 to 2 years of age. Stock was 350 to 510, so 3 to 5 years of age. Preferred were individuals 510 to 710, which was 6 to 8. Quality was 710 to 860, that was 9 to 13. Memorable was 860 to 1020, so about 14 to 22 years in age. And then trophy was anything greater than 1020 millimeters, so anybody greater than 23 years of age. How I did the actual length limits, I broke it into 3 groups. First is the minimum length. So any individual in the green is going to be safe from any fishing mortality. So in the 15-inch minimum length limit, ages 1 to 3 were protected. In the 20, that would be ages 1 to 5, 25, 1 to 7, and the 30-inch was 1 to 10. For the maximum length limit, or reverse it. So now if you're older, bigger, you're going to be protected. We're allowing harvest on only the smaller individuals. So under the 15-inch, 4 to 30 were protected. The 20-inch was 6 to 30, 25 was 8 to 30, and the 30 was 11 to 30. Finally, we have the slot length limit. So I'm allowing harvest on the smaller individuals in the population and larger, but I'm protecting a specific size and age range. So under the slot, 10 to 25 inches, 2 to 7 were protected. Under the 15, 3 to 10 were protected. And under the 20 to 35, 5 to 14 were protected. The last parameter that I needed to estimate was the age-specific birth rate. So how I did this was I generated an age length key using a von Bernoulli growth curve, combined it with a length-weight relationship from Perry Carver 1977, and then that helps scale it to age-specific fecundity, where to get the actual estimate, I used the mean fecundity estimate from Montague and Shoup. I also set the Poisson factor, which was just the reproductive variance, for 1 for all ages. So we've gone through the fun part. Let's go to the actual fun part of results. So for my SIBSHIP results, ML Relate recovered more relationships than Colony, but really all I'm focused on is the overlap number here. So 54 relationships for full siblings were found between both programs. 418 were found between both programs for half siblings. My NE estimator estimate, then I used that for my NE estimate. And my NE estimator estimate was 76.6 from chapter 2. The estimate that I recovered from the Waples and Faraday equation was 75.1, deemed to be the analysis to be excess in this particular sense. We look at the parentage analysis. I found that there were more females reproducing than males, and more females were identified to specific individuals than males. If we look at the mean and variance of these estimates or of reproductive output, on average, each parent is producing roughly 3 offspring. But if we look at the variance, males had a higher reproductive variance than females. All right, now let's look and review the length limit results that I recovered. So from the Age and E result, Some of the higher total N estimates came under the minimum length limit. And as I increased the minimum length limit, the total population size increased, the effective population size estimate increased, my NE to N ratio increased, and the generation length increased. So as I implemented a larger and larger minimum length limit, on average, more older individuals were going to be reproducing within the population, but I'm seeing a more increased overdispersion of reproductive variance. If we look at the size distribution now, under the minimum length limits, I recovered individuals and were estimated to have juveniles, everything up to memorable, but no trophy-sized individuals. And increases in the overall minimum length limit resulted in increases in the number of memorable size fish in that age class. For maximum size limits, I actually recovered the lower estimates of total population size. So as I increased to the maximum length limit, the total population size decreased, the effective population size decreased, the NE to N ratio decreased, and the generation length decreased. So I have fewer individuals, I have very much younger individuals reproducing, and I have a decreased overdispersion of reproductive variance under the maximum length limit. Now, interestingly, within the maximum length limit, if we look at the size distribution, this is the only length limit that resulted in the estimation of trophy-sized individuals being in this particular population. But if we look at the 15 and compare it down to 30, we actually see a decrease in the number of trophy and memorable sized individuals within the population. Finally, let's look at the slot length limit. So the slot length limit resulted in more of a happy medium in terms of overall population size, but by shifting it to incorporate and protect older adults, I saw decreases in the total population, effective population size. My NE to N ratio decreased, my generation length increased. So as I shifted to incorporate older adults, I have now more older adults present in that breeding population, but I have a decrease in the overdispersion of reproductive variance. Under the slot length limit, just like my minimum, I did not recover any individuals in the trophy size category, but as I shifted that slot length limit to protect larger fish, I actually found more fish were estimated to be in the memorable size category. So let's cover this real quick in our comparisons. If per se we want to have the largest population and the largest effective population size, so a little bit more better genetic diversity present, we can go with a minimum length limit. Preferably, you can go with your 30-inch because these were the two highest estimates that I recovered. For the lowest NE to N ratio, so lowest overdispersion of reproductive variance, we would go with the 20 to 35-inch slot length limit. If we want the longest generation length, so we have more of these older individuals present, combined with more memorable and trophy-sized individuals, we would go with the maximum size limit. Finally, we'll cover it just really quickly. Fishing mortality, no surprise, as we increase that fishing mortality, as we add on that extra level of mortality to this population, we see declines across the board in total population size, effective population, generation length. Interestingly, the NE to N ratio, more than likely that's just due to the fact that because the population is becoming so small, we have very few individuals that are going to be possible breeders within it, and that's why I see a reduction in that overdispersion of reproductive variance. And again, on the size distribution, comes as no surprise, increases in fishing mortality resulted in much fewer memorable and trophy size. Interestingly, though, if we look at 22, now we're starting to lose, and onwards, we're starting to lose our preferred and even quality-sized individuals. So more than likely, very bad sign there, and it's a sign of we're seeing that overharvest occurring in as little as, say, 22% fishing mortality. So let's wrap up the last chapter here. Some of the conclusion we can draw is, first, males had a higher reproductive variance than females. We know at the moment that olivaris does provide some parental care. We've seen in other species, be it channel catfish, brown bullhead, or mad toms, that males are predominantly the ones that are providing that parental care. But there's little work that's been done on the reproductive behaviors of olivaris. We don't know whether or not both sexes are playing a role, which sex is going to be the main one that's guarding those nests, things such as that are very basic. We don't really have a great understanding of at the current moment. If we think about it now in terms of the length limits and now apply it for our fisheries, the 15-inch resulted in the higher N and NE estimates. So if we were looking for that sort of great genetic diversity and reaching those IUCN guidelines, we can go down this route. This has also been supported by other studies in Iowa and Alabama, though they mentioned that they would prefer they recommended the 25-inch minimum length limit there. For the 15-inch maximum length limit, we could go that route. Yes, it had reductions in the overall population and effective population size, but it was the only length limits that resulted in memorable and trophy sized. And if we think about it within these medium reservoirs, shifting the minimum will simply shift over the actual like peak that we will see here. Maximum will do it in reverse. For fishing mortality, this study supports the previous work from Winters and Mullen that overfishing was going to occur at around 25 to 27%. I saw my greatest declines after 22%, once I got to 32 and 42% fishing mortality. So controlling fishing mortality is going to be much more important than length limits and actually preserving those memorable and trophy-sized individuals in these reservoirs. What this means for maybe our medium and large size reservoirs is standardizing the bag limit across reservoirs. We have some that are 10, some that are 5, maybe implementing the actual statewide bag limit to limit should help decrease the overall fishing mortality and hopefully start to see more individuals in these larger categories. For smaller reservoirs, it'll be a little bit more interesting because there is the potential that they might not all be experiencing the same fishing mortality because they are smaller and they might not be located in areas that are going to have lots of people fishing them. So before really implementing any sort of bag limit or length limits, it may be best to conduct some creel surveys to understand how many people are utilizing that fishery before making management decisions on the actual fishery first. So with that, I have a bunch of people to start thanking. Dr. Heiss for bringing me here, and then his assistants and overseeing my entire project. Could not have done it without him. Dr. Leah Berkman, Zach Ford, and just all the MDC biologists. I had like 1,000+ genetic samples. There's no way I could have collected them, done any of it, and without them. My committee members, Dr. Anderson, Dr. Garvey, and Dr. Whitledge, for their assistance, their recommendations on this particular project. They've gotten me through 5 years as well. I thank them so much. And I got to thank my past and current lab mates, Dr. Ricky Flamio, Dr. Tiffany Atherton, Juneman, and Tiriana Ross for their assistance in my lab work, helping me out, getting my primers started, some of the PCR work, and even in Tiriana's case, identifying some microsatellites for me. Could not have done it without them. And then lastly, as I come to the end here, I got to thank everybody who's just been on this journey with me for the better part of like kind of now like 12 or 13 years. So from my family to my wonderful wife, Amy, to even my goofball friend Kayla here, even when I was being an absolute moron, to even my best buddy Jacob and all my groomsmen. I also got to thank everybody that I've also had the fortune of meeting here in my grad journey, be it my old advisor in my lab, and everybody here from Journal Club when we did our goofy little prom that was wonderful, to my River to River running group. I could not have made it without any of y'all for the last 5 years. And I'll take any questions you have at this time.